Dr. Melody Bomon is a wildlife veterinarian based in Laos. She's working with the Lao Conservation Trust for Wildlife and the Elephant Conservation Center to provide veterinary care to wildlife in need, survey wildlife disease, and assist in conservation research through the WILDMED project. Being a strong advocate for collaboration, transparency, and knowledge sharing, she is active in projects such as the Gibbon Health Initiative, the Lao Wildlife Laboratory Network, and the Wildlife Medicine Module. So today's presentation is on the role of wildlife rescue centers in wildlife disease investigation. So I would like to invite Dr. Beaumont to start her presentation.
Hello, everyone. Can you see my screen?
Yes, yes, we can.
Okay, excellent. So thank you so much for that introduction. So I'll skip this slide where I introduce myself. But yeah, I work in Laos. I work on different projects, and our main activity is wildlife rehabilitation. Of course, with a conservation focus, but always the primary activity we do is rehabilitating injured, diseased, or displaced wildlife and trying to get them back out into the wild. And I'm sure everyone is aware there are arguments against wildlife rehabilitation. And one of the main ones you will hear is that it's a lot of resources invested in individual animals, and those individual animals don't necessarily have a high conservation value. And another one of the arguments is if it's ethical to bring these animals into captivity, even if it's temporarily. But there's also a lot of arguments for wildlife rehabilitation. And first, that we have a moral responsibility, as a lot of the damage is done by human activity. Second, that individuals do matter when we are talking about endangered or critically endangered species. Or then a big thing wildlife rehabilitation centers can do is education. There needs to be a place for the public to call, to resort to when they find injured wildlife, when they are concerned. The public members that are concerned about this issue need a place to go. The government, the authorities need a place to take the animals when they enforce the law. So it is important that rehab centers exist. And I'm not even starting to talk about all the education projects and potential that is there. But one major thing I want to focus on today is the access we have to wildlife that almost nobody else has. How many people get a wild gibbon on their table or a pangolin? To do research on these animals, it's very difficult. To capture a wild gibbon is very difficult. To all the logistics, the resources, the permits, the ethical concerns. There's so many barriers. But in rescue centers, in the setting we work, we get these animals through our center every day. And there's so much potential. We can detect factors that are outside of the animals, so problem factors. If we get a lot of electrocutions, we are able to identify that that's causing harm to that species. We may be able to identify environmental aspects or social aspects that are impacting wildlife. But we also have access to the individual itself. We can collect samples, we can do disease surveillance, we can do genetic studies. There's so much potential. And if we look at ZIMS, I don't know if you have heard about it. It's a software used by a lot of zoos internationally, and more rescue centers are using it. And here are some statistics from ZIMS that all the animals that are currently recorded in ZIMS institution, most of them are least concern. But if you look at LCTW, this rescue center in Laos, most of the animals they hold are critically endangered. If we look at species enzymes, most of species, so the dark parts of the bars, are not at risk by IUCN classification. If we look at LCTW, the majority of the species they have are at risk. So rescue centers do get a lot of animals that have conservation value. And then if we dig a bit deeper, this is again an extract from ZIMS. If we look at this species, the Lao langur, LCTW is the only place in the world or in within ZIMS users that holds this species in captivity. It means there's virtually no information on this species out there. So we have such a unique access and so much potential to detect um, to collect so much information. And if we look at clinical cases only, so passive disease surveillance, we already detect a lot. This is an avian influenza case in a civet, so it's a spillover event. It's very significant. This small jackal was found in a protected area, but he died of distemper. Uh, Mentioned earlier, it's a concern for wildlife. We've detected melioidosis cases, we've detected drug-resistant bacteria, and this is only on clinical cases. So this is passive disease surveillance. But there's so much more potential that we're still not tapping into, and a lot of centers are not tapping into. I would say more than so much potential, there's so much wasted potential. And then we have to ask why. The answer is very easy. We don't have money, we don't have time, we don't have the resources, facilities. Let's say we have to freeze samples. It's rare that people have access to a freezer or stable electricity. And in the end, we are veterinarians. Our main job is to treat the animal, and our main goal working in rescue centers is that we're doing the treatment and rehabilitation of these animals. Our We're not primarily scientists, we're not primarily epidemiologists, and we cannot be everything. But there is a lot we can do. So first, let's look how we can maximize data collection without investing a lot of resources. Just in our daily clinical practice, what can we do? One thing we can do is sample banking. And this is something we are doing currently at every project I work with. Every time we have an animal and we take a blood sample, we freeze some serum. It takes 5 minutes and we have it. And over the years, we've accumulated hundreds of serum samples. If you don't have a freezer, you can do dried blood spots. You don't need a freezer, you just need filter paper. You put a drop of blood. These can be kept, as long as they stay dry, they can be kept up to 5 years. They can be used for disease surveillance in the future. So much potential for genetics. You can do hair samples. Hair samples can be used for, again, genetics. They can be used for um, pollution, uh, how do you say it? Pesticide detection, for endocrinology. Don't need a fridge. Takes 10 seconds to collect a hair sample, pop it in a bag. And there's tissue samples. Formalin doesn't need a freezer. Solutions like RNA laser are really expensive, but we can do this with low resources. Just alcohol 70%, ethanol 70%. A tissue sample stored in alcohol can be used for so many purposes, from genetic studies to disease surveillance. There's so much potential. And then, not speaking specifically about disease, about infectious disease, there's so much data we can record. So we can record sex, color patterns, dental records, biometrics. And in all of this, one really, really, really important point is we need to be consistent with how we collect this data. If we're going to collect samples, if we're going to collect data, it needs to be done properly. If we collect a measurement and every time it's measured, it's in a different way, then that data is useless. So it's really important to have protocols. And this is something you make once and then you can just apply it. So we take biometric data on every primate and we do it the same way every time. And we have these very detailed instructions. So if different vets or different staff members take the measurements, they will use the same landmarks. They will take the same measurements. And this goes for everything. These are our instructions for sample storage. We have instructions on how to label, what information to place on the label, because again, we can collect the sample, we can collect it properly, and then if we don't label it properly, if we're not able to identify where that sample came from, it's worthless. It's really important to have all these details lined out and make sure that the whole team is trained on this. This is an example of a necrocy protocol for elephants. So it's we can use other protocols to inspire us and decide what we want to do and what is possible for us to do. So this protocol has 3 pages of suggested organs to sample for histopathology. But of course, not everyone can do that. That is very time consuming. But we can say, us as a center, every time an elephant dies, we will collect liver, kidney, spleen, lung, and heart, 5 samples. It takes 15 minutes, pop them in formalin, they go into the sample bank. So those are all things we can do. These are some of the pictures we take. So these are identification pictures. We always take them for every animal that goes through a health check. We take these standardized pictures, and it may seem that they're not very useful, but in the long run, they are. And this is, again, something very quick, takes no time, takes no resources, you just need a phone. And if you compare here, this is the importance of doing it properly and always in the same way. That first picture cannot really be used, not for identification, not for any color patterns, or it's at a weird angle. The animal seems longer than it is in real life. But that second picture, that's a good picture. And then we've now reached the stage where we have enough pictures where we have, for example, dental progression. We can see dental wear. We can see the sequence of eruption in different species. We always take genital pictures, which may seem really weird, but it's surprisingly hard to sex some species, and there is very little information out there. But now we're forming this database of genital pictures, and we can use this to then train new staff members, share with other centers on how to properly sex the animals. So these are very simple things that can have a lot of impact, and they don't require a lot of time. They don't require a lot of resources. Something that is really, really important is record keeping. Let's say we take these pictures and then they are on somebody's phone and then that person leaves. We took them for nothing. We need to know where things are. We need to make sure they're recorded properly, that we can look back at it. So things we need to think about when we decide on record keeping is what language we're recording in. Are we doing paper files? Are we doing digital? Are we using any software? You can just do Excel databases or Google Sheets. There's software like Zims that is really useful. What is important is that it's clear, it's consistent, and it's really nice when it's searchable. And again, you don't need very fancy tool to make something searchable, because of course Zims is amazing and I highly recommend it, but it is not free. But things like Google Drive are free. So this is how we keep our pictures. Every time an animal gets on the table, pictures get taken. Whoever took the pictures uploads them onto the animal's file. And you see here we have rules for animal pictures. So it's again standardizing things. And these rules specify that every folder has the animal ID number, then the scientific name, and then a house name or any relevant identifying features. And why is this important? Because this creates a searchable database. So now if I search Mamascus, I see all my gibbons. If I search for gibbon genitalia, I see all my gibbon genital pictures, and so on. So we're creating a searchable database of relevant information on species where there's very little information out there with very little resources. But what now? As I said earlier, I have hundreds of serum samples in my freezer. I still don't have time. I don't have a second freezer. I'm not a researcher or an epidemiologist. It's not my primary goal. And for some species, I do have very small amounts of samples. There's not much scientifically relevant that I can do with 5 uh, ferret badger serum samples. It's not a large enough number. So what else is really important? Is collaboration. And I know this started to sound like a TED talk, but it really, really is. And I want to talk about Laos as an example. So there's not many organizations working in conservation. So on this side of the screen, these are organizations working in wildlife rescue. On the other side, you see some organizations that are involved in conservation and more field work, policy making, forensics. And at one point, we all sat together and we created the Wildlife Health Laboratory Network. And it's very informal. And basically, we just sat together and we decided to communicate, be transparent, share resources, and try to take actions that would last for the long run. So not do a short project, a 3-month project, and then when the funding ends, it never continues. We're trying to do things that will last. So we had a meeting where we looked at what we do, what we have, what capacity do we have, what capacity do we want, and what is priority. We identified the capacity that we had in country. This is 2023, and it was very, 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 very little. We had almost no labs to do animal disease specific testing. We couldn't look at any turtle or tortoise pathogens or even primate diseases, if only if shared with humans. There had very, very little capacity. But the second thing we did was look at what each of us wants to focus on. And we all have different areas we're looking to develop. And for example, you see the ECC, they have an endocrinology lab. It doesn't make sense to make a second endocrinology lab in Laos because even the one at the ECC right now, it's not being used 100% of the time. So it doesn't make sense to duplicate resources. It makes sense to share resources. And that's what we're trying to do. So we share what we have. We all focus on different areas, and that way we're developing faster. And we also are now working on a joint sample bank. So what I was talking about earlier about only having 5 samples of one species, I may have 5, but Free the Bears may have 7, and WILDMED or the ECC can have 10. And suddenly we have a bigger number. So what we're doing now is we're joining our protocols and our SOPs. We're all taking the same samples. That's the goal, that we will all take the same samples in the same way. We will store them the same way, and then we can share them for different research projects. And this all sounds very nice, right, in theory, but does it work? It does work. We've achieved so much since we started collaborating more. So this is an example. It's genetics, but I think it's a really good example of the collaboration. So we had these crocodiles, 85 crocodiles, and we did not know if they were hybrids or if they were pure Siamese. If they were pure Siamese, they're very important in terms of conservation, but if they're hybrids, they're not. And there was no capacity in country to do the genetic testing. Exporting the samples with CITES is currently very, very difficult because of the situation in Laos. And for years, we've been wanting to test these crocodiles. So what did we do? We collected the samples two organizations collected the samples. Now we stored them in this -80 freezer from a different partner because we don't have one, but they have one and they had free space. Then IRD, another partner that are more focused on lab work, they helped with the DNA extraction, the lab space. And then we had an external partner to do all the bioinformatics and everything. So by joining forces, we were able to determine that 88% of our crocodiles were pure Siamese. We were able to separate out the individuals that were hybrids, and we now have a very successful breeding program where we're hatching these baby crocodiles. They're being released back into the wild. So we made it, and this is really great. And we have many more examples. This is not the only one. IRD with the ECC did a study on non-invasive TB screening. At LCTW, we collect samples. So every animal that came through for more than a year, every mammal would get nasal, oral, rectal swabs. It took 1 minute to take this. And then we would just send them off to IRD, and they did all the lab stuff, the storage, the testing. So that way we were able to do COVID-19 surveillance in wild mammals in hundreds of samples. And it took very little of our resources, of our time. With a malaria study in fecal samples, we're keeping ticks. So any tick I find, I pop them in alcohol, we keep them, and then we send them off to this laboratory partner. There have been Asian elephant genetic population surveys with these partnerships, endocrinology studies, currently working on gibbon speciation. So again, samples are coming from one place, another place is doing the sample processing, and then we send it off externally. So I think this is a really good example of how you can do so much with very little resources. And we work in NGOs, we work as nonprofits. There is no competition. We're all working for the same thing. We're all working for the same goals. We're not competing over anything. And when we work together, we can achieve so much more and so much faster. So we're fully transparent on what we're trying to develop to make sure another partner of the network doesn't develop the same capacity. So we're not wasting resources. We share what we have. So if we have capacity, we don't keep it just for ourselves. We share it with every other partner in the network. We apply for funding together, and it's actually more successful to get funding awarded when you apply together with multiple organizations. And we're really focusing on establishing capacity that is going to last. And this has led not only in terms of laboratory and disease investigation, but this collaboration is becoming more and more solid. And we've done other projects as well. So we've done, IRD did a parasitology training where multiple of the members attended and benefited from. And this is one of my favorite projects, is we developed a wildlife medicine module between the three rescue centers in the country with the National University. It's now part of the curriculum, which means every veterinary student of the country goes through this program. And it's a 2-week intensive program where they get hands-on practical classes on wildlife, on conservation. Here you can see a class on crocodiles. We go through necropsy, One Health, biosecurity, and we've actually hired already 4 people out of this program are now working in conservation, 3 of them in rescue centers, and 1 of them with the One Health project at WCS. So it's really successful, and it's just so great to be able to do this. So yeah, I think the take-home message for me is focus on what you can do, not on what you can't do. Because there is a lot we can do, even if there is no lab and there is no time and there is no money. There's so much we can do. And the main thing to make it work is to be organized, have protocols in place, do things consistently the same way, proper record keeping, and collaborate, talk to each other, to get more capacity, to join forces to be able to achieve these goals. And this is not, I gave this example about Laos, and I think we're really doing great work, especially considering how limited our resources are. But this also happens on a regional level, and I'm just going to pitch the Gibbon Health Initiative. So it's run by VIEW, funded by ARCAS, and the goal is it's the same thing. It's to join all rescue centers, all in situ centers that work with gibbons, and communicate, share resources, share knowledge. So if anyone works with gibbons and is not yet part of the network, please reach out, because we would be happy to have you join. And yeah, I think that's all for today. Thank you very much. I hope this was useful.
Yeah, thank you, Dr. Melody Bomon, for the wonderful talk. It's a great work that you are doing at Lao Conservation Trust for Wildlife and also ECC. And I have one question, that is, I would like to just know more about ZIMS, throw some lights on ZIMS and what are the basic, how does it work? That will help us to know more about ZIMS. Yeah.
Okay, I can share my screen again and give you a quick walkthrough. So I'll just log in. Yeah. Sometimes a little bit slow to log in. So basically, it's an online database. It's designed for wildlife or captive wildlife, and they are now expanding to include also rehabilitation, post-release monitoring, and all of that. And let me just log in. So the big advantage is that all of your information is in the same place. And so I can see all my animals here. They're very cute loading screens. Let me just go. Just 1 second. So you put all of your animal files in here and you can record any information that you can imagine. ZIMS has a place for it. So I'm going to put Calypso. I'll just pull up a random record. My internet's been very slow. So this is the animals file. The microchips in here, all the biometrical measurements we took are in here. There's weights in here. Then you can create a weight graph, so you can see the animal's weight evolution. You can compare weight evolution of different animals. You can see all the enclosures they've been in. Then there's medical records that are also very detailed, where there's separate fields for anesthesia prescriptions, if there are any adverse effects or anything, lab results, everything goes in there. And what is really great is that all this information will contribute to worldwide reference values. So while this loads, there's these resources, so medical resources. So I can look at drug usage extracts. So let's say I want to see, can I give cimetidine to an elephant and what is the dosage? I can actually check on ZIMS and see if people have used it, what dosage, if there have been any adverse effects. And so let's see, wait, LFS Maximus. So again, it's all about data sharing. None of the records, it doesn't link back to your institution, so nobody will be able to say, oh, that specific place they gave cimetidine and the animal died. But what you get is sort of a statistical overview. So I can see here cimetidine was given orally, at most people are giving around 2 milligrams per kilogram. And then if there were any side effects, he would show that in different colors. So that way I can see the dose range that people have been using, the route they have been using, if twice a day, if once a day. If there's any adverse effects, it will show up. The same way you can look up test results. So I can look, let's do again, elephant. And for Asian elephant, let's do Indian elephant. So you can actually look at reference ranges based on the data that all the ZIMS institutions are entering. So here you have a red blood cell count range based on 2,500 samples. Obviously, it always needs to be used in a critical way because this data is not being validated and there is a certain degree of error, but it is very useful, especially when you're working with species where there's no published information. And you can also filter this, so you can filter this to females only or to only the continent you're working in or the country you're working in. We have hematology, chemistry, all of that. So I showed you, so these are the resources. This is the animal records, and there's space for observations, any behavioral observations, anything like that. Then the medical records. So for example, here for this animal, on this day, she had an anesthesia event. Everything about the anesthesia will be here. All the medication she got, the dosages, if there were any adverse effects, anything like that. Any samples are here. The results of the testing, if the samples were tested, are in here. Any treatments, everything goes in, just like medical records anywhere else. The only difference is that the data is going to contribute to this worldwide pool of data. And then another module that I find very useful is the sample storage module. So actually from the medical records, let's say I take a sample, I can move this sample to storage, you see here, and then within ZIMS, I can have a sample storage, so a biobank database. So I can actually look up I can look up, let's do a Gibbon, any Gibbons, any samples. Let's see which Gibbon samples we have in storage. So now I can see all the Gibbon samples I have in storage, the type of sample, I can see where they are. So I have this kidney sample that is in the storage cabinet in the hospital. So you can have your whole sample data bank. You can just, it's a big database, and it's very useful. And yeah, was that good as a introduction?
Yeah, great, wonderful. How to access it? Is there any username and password? How to be a member of it?
So it is paid, which is the downside. It is run by an NGO, but it is paid. They do have a sliding scale. So for organizations in developing countries that are non-profits, the annual price is much, much, much lower than for a big zoo in the United States, for example. So it's basically emailing them, and they'll ask a set of questions and then tell you in which category you fall for the price scale.
Thank you, Melody. Heidi has a question, I think. Dr. Heidi?
Yes. Yep. Hi, Melody. I have a question specifically about the TB testing that you do with the elephants at the center, ECC. So what type of testing do you do? And you test just the elephants or also the mahouts or the staff?
At this moment, so the study I referred to was a pilot study to trial non-invasive testing. So it was with fecal samples. The thing is, we didn't have any positives, so we need some positive cases to trial if it would even work. But at the moment, we're not doing any routine TB screening. Yeah, we aim to, but we don't have the full capacity yet to do so.
Okay. So you don't do like trunk wash cultures or things like that. So you tried to do the fecal, did you do cultures?
We did, yeah, but it was a one-time study. Yeah.
Okay.
And
so did you do just the elephants or also the mahouts?
The elephants and the mahouts.
Okay.
Yeah.
Is there any TB screening process in Laos for people where the mahouts could kind of come under that particular screening process?
There is a program that does nationwide screening with GeneXpert, but you have to fit the criteria. So you have to have certain symptoms to be eligible for testing. The other option would be chest X-rays, or we have a way of doing the skin test. So it's definitely it's something we're working on. We're hoping to, by the end of this year, to start to do it regularly.
Okay. Okay. Thank you.
There's a question from Dr. Palash. How to join a program at Laos? That's a question.
What do you mean to join as in a visitor, as a
maybe as a volunteer, maybe
or to join the network?
Volunteering or as an intern in any of the programs that you have?
Yeah. They just reach out to us, email us. We're all happy to host people and to share knowledge. I'll leave my email in the chat as well, and feel free to email me.
Okay. Thank you. Any other questions for Dr. Melody? Yeah. If there is any question, if there is any more question, maybe we'll email you, and we can just circulate with all the members of the group. Yeah. Thank you, Dr. Melody, for the wonderful presentation. It's a great work that you're just doing there in Laos. And once again, I just thank you on behalf of Asian Elephant Trust and Life Trust India. Thank you. Now over to Heidi.
Yeah, thank you so much for inviting me, for having me.
Yeah, thank you. It was very interesting. Very interesting. Great work you're doing. So.